Assalamu alaikum wa rahmatullahi wa barakatuhu, and a very good morning. So, how are you all? Okay. So, may I just know your levels in biology, like A, B, C, S, O, W? Like, be frank, okay? Just, I want to know your levels. Okay, there are S, B, and also C. Okay, W is also there. Okay, nice. So this revision program was planned for your year 27. Yeah, for both. This can be for both 27 and also for 26 because the first four units are important for both the batches. No? So both of y'all can join and get the maximum use. So I just give you all a brief idea of what we are supposed to do in this. So during the session, yesterday also I sent a message in the group like what you all are supposed to do before the session and during the session and after the session. So the first thing you all have to do before the session is either you can go through our short notes, like today we are doing the first four units. So the first tutor is the intro, and the second one is cell crash, and the third one is Evo dive, and the fourth one is Flora flash. So you just go through those tools or else through the resource book for the first four units because you need a revision. And then during the session, what you're supposed to do is we'll be like touching each and every part of the theory, like every single part of these first four units today, inshallah. So and then I'll be giving certain MCQs, like the selected MCQs from the whole tool, because today we have 355 MCQs and it's not practical to do all the questions once. So, like within these 3.5 hours. Therefore, you are supposed to do all like the selected questions today, and the remaining questions you all have to finish them within today. Okay, that's why the revision is planned for you all. So, after finishing them, I'll be uploading the scheme tomorrow morning at 5:30, inshallah, and then go through the scheme and y'all can correct your own mistakes. And after that, I hope y'all got the biocheck tute. Is that so? Y'all got all the biocheck tutes, no? All the 10 tutes were with y'all, ne? Yeah. Okay, so y'all can try the biocheck tools after you're done with the MCQ questions. Okay, because that is to like check your statement things and to be more accurate in essays and structured essays. So after that, we gave a guessing essay thing for 26 batch. So y'all can refer the first four units in that and try to write maximum essays as possible from those parts. Yeah, we'll be releasing the biocheck schemes. Like for all 10, we'll be releasing the schemes. That's also most probably by today or tomorrow. So after that, go through all the essays and try to write maximum essays as possible. Don't have any reasons right now because you all have only 15 days now. So yeah, try to write more and more essays. So most probably the session would be around 3 to 3.5 hours. So yeah, let's start the session. So from the beginning we'll go. Like first we'll go to the introduction of biology, the first unit, and just keep this session more interactive. You all also have to talk, and yeah, it's not only me who talks. So first we are going to the introduction of biology. So for the biology there is a definition. What is biology means? It's focused on studying living organisms. Okay. And then we have the three primary branches. Remember, we have the three primary branches. The three primary branches are, these are primary branches. Okay. Zoology, botany, and microbiology. These three are the primary branches. And then we have the areas to study the biology. Okay. Yeah, areas to study the biology. So we have uh, these things: cell biology, histology, anatomy, physiology, biochemistry, genetics, and ecology. After that, we are having the issues pertaining to biology. Okay, so we have 8 issues. So that the first one is we have to understand the biological diversity. Among that, we have an important point about the life formed on Earth. It is what, 3.5 billion years ago. And the first formed organism is having three main characters. This was like questioned in many MCQ papers. So the three main characters are heterotrophic, anaerobic, and prokaryotic. Okay. And also, there are about 10 million to over 100 million species. Okay. Remember, 10 million to over 100 million species in the world. And then there is a dynamic relationship, not a static relationship. These are like keywords: dynamic relationship between the living world and inanimate world. And also, biodiversity. Okay, remember, biodiversity includes three main things. Okay, first is the number of species of plants, animals, and microbes. And the diversity. Second one is diversity of genes. And the third one is what? Diverse, different ecosystems, diversity of ecosystems. So these three are the main three varieties of life, like varieties of life depends on these three main things: number of species of plants, animals, and microbes, diversity of genes, and different ecosystems. Second one is, see here, the human body and its function. So to study about the human body, we have to study histology and anatomy. Tell me soon, what is histology and what is anatomy? What is histology and what is anatomy? Tell soon. What is histology and what is anatomy? Very good. Studying of the tissue is histology. And what is anatomy? Very good. Studying of the gross structure, not the cross structure. It is the studying of the gross structure. So studying histology and anatomy will be able to give a better knowledge about the human body system. So this also can be questioned in MCQs or else in structured essay. And then by this we can understand the human body. And for this also helps to identify the functions of human body, functions of different organs and different systems. And after that, the third main issue is what? Sustainable use management of natural resources and environment. So for the in this part, you have a definition for the natural resources. So natural resources are what? They are materials or energy which are found naturally. Remember, they are found naturally, and they are used in two main things. One, they can be used in everyday life, and then they can be used for what? Economic development. So these all these points have to be included: sources of materials and energy found naturally, used in everyday life, and used for what? Economic development. So all these four main points have to be included when writing an structured essay answer or else for the essay question. And these natural resources are very limited on Earth. And due to the increase in human population and the overuse of these natural resources, these were what? These were depleted. Okay. So the depletion of natural resources are due to what? Due to the increase in human population growth, and also what? Due to the overuse of natural resources. So these two are the main reasons why the depletion of natural resources are taking place. And then see here, these are what? These are the environmental problems which are caused due to the over exploitation. Environmental pollution, loss of biodiversity, desertification. These three are the main three environmental problems caused due to overexploitation. This was also questioned in many structured essay questions. So remember this part. And then we have the fourth one, sustainable food production. So here again, we are having another definition. This is also an important definition for the sustainable food production. Sustainable food production means we are like producing amount of like a certain sufficient adequate amount of like the amount which is required by our body. So we are producing only an adequate or sufficient amount of food. Why do we produce how do we produce these things? By using environmentally safe methods. You have to include both these keywords. Okay, we are using environmentally safe methods. Why do we use environmentally safe methods? To produce sufficient amount of food. Okay, so this is the definition for this sustainable food production. And remember, the current population of the human is what? 7 billion, and it was expected to double less than in 40 years, not in 40 years, less than 40 years. Okay, less than 40 years. Those are keywords. Remember that I'm saying this again. And also, there are three methods. Okay, three methods to for the sustainable food production. So they are what? Producing high yielding. Remember these terms only. See here, high yielding varieties of plants and animals. Producing disease resistant varieties of plants and animals. The first part and the last part are the same. Only the middle part is changing. So remember high yielding, disease resistant. And also post harvest methods, the harvesting things after the processes after the harvesting parts. So this also have to be increased to have a sustainable food production. And also we have to understand what? Plant life. Why do we supposed to understand the plant life? Because plants are what? They are the primary producers on earth. And also to understand the plant biology, we by understanding plant biology we can produce two types of plants. One, they can be high yielding, and the second one, they can be what? Disease resistant. And then we have certain diseases like cancer, AIDS, heart disease, chronic disease, and dengue. And these things they have classified them as what? Communicable and non-communicable diseases. And for the cancers, they haven't understood the causes still yet. And also AIDS is a growing problem. Heart disease is also another growing problem. And CKDU, the chronic kidney disease of unknown etiology, is a serious problem in Sri Lanka. Okay, CKDU, we have two main types. One is CKDU and the other one is CKD. So remember, CKDU is a growing serious problem. And again, dengue is a growing problem, a serious problem. And then we have the solving legal and ethical issues. So here we have three main uses of solving legal and ethical issues: parentage testing, criminal investigation, and solving immigration disputes. And for this, we are mainly using what? Fingerprinting. And then, yeah, this is just a flow chart. And you all can go for the characteristic of organisms. So there are like four characteristics. In characteristic, you can remember these mnemonics. Oh my god, I am really hungry. O, organization, odd and organization. M, metabolism. God, growth and development. I, irritability and coordination. A, adaptation. R, reproduction. And H is hereditary and evolution. So this would help you all to remember these eight characteristic of living organisms. And even though all these have eight characters, they either show these characters simultaneously or at some point of life. Okay, remember, can show simultaneously or else at some point of life. They do not show all of them at one time. And also, some non-living organisms such as crystals have certain living characters. So crystals can grow and waves can move. So growth is not only for the living organisms, can also be for both non-living organism. And we have single-celled organisms and also multicellular organisms. We'll be studying that deeply in unit 2. So in order in organization, remember from molecular level, okay, from molecular level to biosphere, there is an order in organization, okay, and organisms are organized from lower level components to upper level, not from the upper to lower. It should be what? Lower level to upper level. So they are organized from what? Lower level components to upper levels. That's how the plant leaves, human eyes, each and every part of the organism was organized. And metabolism. Second one is what? Metabolism. It's the sum of all chemical activities taking place inside an organism. All chemical activities, okay? Chemical activities taking place inside the organism. And we can divide this into two main things, catabolism and anabolism. Catabolism is breaking down of complex molecules. Anabolism is what? Building up of molecules. So for this, you have these two things, no, absorbing energy and releasing energy. So remember, anabolism is starting with ana. So anabolism absorbs energy. Catabolism is what? Releasing energy. And the third one is growth and development. So growth is what? Irreversible increase in dry mass. This term is very important, irreversible. Okay. Remember to add this term whenever you write the definition, irreversible increase in the dry mass. And irreversible changes that occurring in the lifespan of an organism is called as what? It is the development. Irreversible changes occurring in the lifespan of an organism is called as the development. And these two, the growth and development, are two consequent processes, not the consecutive processes. Okay, they are consequent processes. Remember this term. This can be questioned for MCQs. And then we have the adaptation. So adaptation is a peculiarity of structure, physiology, or behavior. These three terms have to be included: structure, physiology, and behavior. And also they are promoting what? They are promoting the survival and reproduction of organism. So we have three examples: uncle stomata of xerophytes, viviparity in some wine groups, and splayed output in camels. These three are the main examples for adaptations. So remember to add these things: structure, physiology, and behavior of organisms, which are promoting the survival and reproduction. And also reproduction is producing offsprings. Why do we produce offsprings? For the continuous existence of species. Okay, for the continuous existence of species only we are producing these offsprings, not anything, not a population, not a community or anything. The term species have to be included here. And then we have hereditary and evolution. So genes are passing from generation to generation. That's how the evolution is, sorry, hereditary is taking place. So genes can control for three main characters. One, they can control the physiological character, or else the morphological character, or else the behavioral character. So these four characters are controlled mainly by what? Mainly by the genes. And also evolution, we have the definition for evolution. It is the ability of the organisms to change over a long period of time. Okay, and this is occurring as a result of what? Due to the genetic modification. And finally, we have the irritability and coordination. So what is irritability? It is the ability to respond to stimuli from both internal and external environment. Have to remember this term: both internal and external environment. If they have given only one term, again that statement is wrong. Okay, even when you're writing the structureless equation, have to include both the terms: respond to stimuli from both internal and external environment. And then movement of organisms mainly occurs due to as a result of both irritability and coordination, not only one. Okay, both irritability and coordination have to take place for the movement to occur. And also this coordination is mainly involving in four main systems: nervous, hormonal, muscular, and skeletal. So remember these parts as well. So here through this chart you all can refer the characters of living organisms and non-living organisms. And I hope you all know the hierarchical level of organizations. Ne? So they are going from molecules, organelles, and remember cell is the basic structural and functional unit of life, and it is the living one. Okay, in the hierarchical organization, the living stage is starting from the cell. After that, it goes tissues, and then finally organs, systems, organisms, and then population, community. That's how the ecosystem, and finally it goes to biosphere. So that's how the characters of, sorry, the organizational levels of living organisms are taking place, and these also you all can like quickly go through. And then, yeah, we have the nature and organization patterns of living organisms. So here, organisms are mainly diverse based on four main characters: size, shape, form, and habitats. Okay, so organism size can vary from the smallest size to the largest size. The smallest size is the 1 micrometer to 5 micrometer of the bacteria, and the largest size is what? The giant sequoia plant, which is 100 meter. And yeah, the shape can be of two types. One, either the cylindrical shape or streamlined body shape. Cylindrical is for earthworm and streamlined for birds and fish. And there are two main forms, unicellular and multicellular. Unicellular means what? Single celled, and multicellular is many cells. And habitat, they can be different habitats: terrestrial, aquatic, arboreal. Arboreal means what? Living on the trees. And then aerial. So these are the characters. uh, that's it about this part. Yeah, they have given all the points now. So now let's go for the revision tute. Okay, revision tute. I want you all to try all the MCQs. There were like around 19 MCQs now. So try out all those MCQs soon. From unit 1, try out all the MCQs as soon as possible. Try them soon. I'll give you all 10 minutes now, 5:56. Before 6, try to do all the MCQs. Have to be very fast. Do all the MCQs soon. We have 16 MCQs, just 16 only, and from unit 1, do those questions soon. No, not all the organelles are living, only the organelles which are like, only the nucleus is living, because the cells which consist of nucleus is living. The cells which do not have nucleus is non-living. And not all the organelles are consist of nucleus, because nucleus itself is an organelle. Therefore, the cell is the basic living structure. If you all have any doubts, ask. Yeah, we'll be providing the recording. Uh, Memorizing only the minutes note. Like, you don't have to, like, if you are a 27 veg student, I don't recommend you all to memorize only the minutes this one minutes note, because you all have to be thorough with the resource book. This was like made based on the resource book, Tama, but there might be human errors, no? So for that, better to have both the resource book and also the minutes tute. I'll anyways upload the tute again to the chat box. If y'all don't have the tute, y'all can try from that. You all got 3 minutes more. Try to do this fast as much as you all can. Yeah, again, I'm saying don't just memorize minitudes, have the resource book as well. But if you're a 26 student, like it's okay to go through the minitude like for now, because if you're like, especially for the last 5 units, okay, because those units are like things which we don't usually study, no, just ignore it. So you all can refer the minitude, but don't just blindly depend on that only because human errors are obviously possible, and if there was any errors like that cannot be corrected so better to have the resource book as well because resource book is the key for biology. And I just made the minute tudes to for the easy reference only because it's like the resource book is always black and white ne? Okay, now it's 6 6, so let's move to the discussion. So first question, which of the following is false regarding the criteria of the organisms? I want you all to say me the answer, okay? So what's the answer for the first question? What's the answer?
Both.
Very good, nice. The answer is 4, cellular organization. Second one, which of the following indicates the hierarchical level of organization of living things in the correct order? Which is giving the correct order? Tell soon. The answer is what? How should it go? Cells, tissues, organ, system, organism, and then from organism we can use the term species, and then from species it is going for the population, community, ecosystem, and biosphere. So the answer is what? Two. Nice. Which of the following feature is correct regarding the importance of physical property, not the chemical property? This is unit 2, no? Sorry, physical property of water for organisms. So we have four main physical properties. Yeah, let's go for the cute and see. See here. These are the four main, like we have both the chemical properties and then physical properties are there. Okay, see here. Chemical properties. What are the chemical properties? It's small, polar, and angular. These are chemical properties. And what is polarity? It is the uneven charge distribution within a molecule. And oxygen is having what? Slightly negative charge, and the hydrogen is having slightly positive. You have to include this term, okay? Slightly is very important. And this, due to this slightly positive and slightly negative charge, it is having a what? Having a polarity. And also, what is hydrogen bonds? Weak attractions. Those are not strong attractions. Those are what? Weak attractions between slightly polar. Again, you have to put this term: slightly polar hydrogen atom of one molecule and slightly polar oxygen atom of the adjacent molecule, not inside the same molecule. And hydrogen bonds are playing a major role in maintaining the chemical property and physical property of the water. And these hydrogen bonds are very fragile. Remember, they are very fragile. In which form? In the liquid form. So they can form, break, and then reform with what? Great frequency. With great frequency. Remember all these terms which I circle. And these are four main physical properties of water. First is cohesive behavior. What is cohesion? Cohesion is the attraction between water molecules, and it is mainly due to what? They are mainly due to hydrogen bonding. And the addition is what? Attraction between water and other substances. And these cohesion and addition together helping the water to act as a transport medium. And mainly these minerals, okay, inside the plants, minerals are transported as a continuous column. Remember, minerals are transported as a continuous column due to the inside what? Inside the xylem, and this is mainly due to cohesion. And addition is helping. Its addition is present between water and other substances. So here the other substance we have is what? Cell walls. So this is helping for the conduction. Okay, conduction of water is supported by the addition, and the flow of the minerals through as a continuous column is supported by what? Supported by the cohesion. And also small organisms like water skaters can walk on the surface of the water molecule due to what? High surface tension of water. And then we have the second main character, ability to moderate temperature. So organisms can absorb and water can absorb and release large amount of heat, right, by a slight change in its own temperature. Like a temperature change would be very small, but still a large amount of heat can be either absorbed or else it can be released. And we have two main terms. One is the high specific heat. Okay, due to high specific heat, water can act as a thermal buffer in both living system and aquatic bodies. Okay, so during temperature fluctuations on earth, water would act as a thermal buffer due to what character? Due to high specific heat. And due to the second character, high heat of vaporization, this water is removing, with the minimum loss of water, it can remove large amount of heat. As a result, it is helping to cool the body surface, and finally this will prevent what? Prevent overheating. And this is the main two processes taking place in organisms. One is evaporation. Through evaporation, what is happening? The human body is becoming cool, and through transpiration, plants are becoming cool. And see here the third character, expansion upon freezing. So remember, water is having the maximum density at 4 degrees Celsius. And whenever the temperature goes below 4 degrees Celsius, water will freeze and form what? Ice. Ice is the crystalline structure of the water. And ice have density lower than the water, therefore this like what floats on top of the water, and the water would be present below the ice particles. And then in during winter and stuff, the organisms can survive, like aquatic organisms can survive under the ice. uh, Ice. This is mainly due to what? Expansion upon freezing. And then we have versatility as a solvent. Here remember, there are three main substances. One is polar molecules, second is ionic molecules, and the third one is polar ionic compounds. So for polar molecules we have glucose, for ionic we have sodium chloride, and for polar ionic we have what? Lysozymes. And what this one, the solubility mainly depends on polarity, not on ionic nature. Remember those things. Now let's go for the question. See here. High specific heat capacity supports the cooling of the body surface of terrestrial animals. Which statement is correct? In the third one, which statement is correct? Which is correct? I want the answer from everyone. Fourth one is anyways wrong. See here. Depends on polarity only, not on ionic nature. And then what is the other option? Three. Go for the three. Expansion upon freezing helps organisms survive during winter. Very good. That's the correct statement. See, first one, let's see why it's not correct. High specific heat capacity supports the cooling of the body surface of terrestrial animals. Is it the high specific heat capacity? Quickly jump here. High specific heat capacity is helping to cool the body of temperature of what? Aquatic bodies mainly. And high heat of vaporization is the one which is mainly helping to cool the body. Conduction of water and dissolved substances through vascular tissue is solely due to cohesive. Is it only due to cohesive forces? No, right? It's due to adhesive forces as well. Low transparency of water helps plants to grow in deep areas of water bodies. Due to the low transparency, like light can penetrate the water, and like aquatic plants can do photosynthesis. Which of the following can be hydrolyzed in human body? What's the answer for the fourth question? Can be hydrolyzed in the human body? Answer. Deoxyribose, lactose, nucleotide, fatty acids, and amino acids. What can be the answer? It is what? It is the lactose. Okay, remember it's lactose. It's lactose because lactose is hydrolyzed by the lactase, and yeah, as a result only what is happening, because lactose is a disaccharide, can be hydrolyzed by lactase in the small intestine, and the other things like we have the nucleotides, fatty acids, amino acids, all of these are what? All of these are monomers, so they cannot be hydrolyzed further. What is the most accurate definition? See here, what is the most accurate definition for sustainable food production? What is the answer? What would be the answer? What is the answer for that? Very good. It's the fifth answer. Okay. In the order of in the organization levels of living matter, in which level plant leaf can be placed? This is a baby question. Like even nursery students can respond this one. What is that? It's the organ, no? First organisms evolved on the earth consist of what are the three main characters of first organism? Tell soon. Three main characters of the first living organism. What are the three main characters? Soon, I want the answer from everyone. Three main characters of the first living organism. Prokaryotes, nice. Very good. Anaerobic and heterotrophic. See here, photophosphorylation. If they are autotrophic only, they can do photophosphorylation, no? So this one is wrong. Eukaryotic, this is obviously wrong, like directly wrong. And then they have RNA, obviously that's correct. Prokaryote, this is correct. Glycolysis. For glycolysis, do they require this one? What is that? Photosynthesis. Is glycolysis required for photosynthesis? No. Even without the uh, even the heterotrophs can do what? Glycolysis and then RNA. So answer is what? 2. Prokaryotic. Again, this is correct. Circular DNA. Do they have circular DNA? The initial organisms? No, right? They had what? RNA. And they didn't had a protein coat. Prokaryotic, correct. Electron transport chain. Electron transport chain is a character of which organism? Which is this is for ETC is present mainly on like can be present in both. This is coming under cellular respiration, but these organisms initially didn't had that much of complex structure. And then enzymes, we had the enzymes such as what? In RNA acted as both enzymes, no? So this is also correct. Plasma membrane was not present. They didn't have ribosomes and also didn't have Calvin cycle. So answer is 2. The threat of depletion of why does the threat of depletion is occurring? Answer for 8. What is the answer for 8? Very good. Overuse of natural resources and also increase in the growth of the rate of human population. Two characteristics that can be seen only in living organisms. Only in living organism. What is the answer for the ninth question? Only in living organisms. Very good. Again, it's the fourth answer, metabolism and heredity. Which of the following definition is most accurate? Most accurate definition. Metabolism is the sum of all chemical activities occurring in a cell. Is it occurring in a cell? Is it occurring in a cell? No. Then where does it occurs? Occurring in a... I want everyone to be active. Don't be lazy in the morning itself. And you have only 15 days. Very good. Occurring in the organisms. Very good. Irritability, responding to stimuli from both internal and external environment. Is it just responding? No, right? It's the ability to respond. Therefore, this is also wrong. Reproduction. It is the process through which a new generation of individual produced from existing organism. One thing is missing here. What is missing? What is the term missing? I said all this thing earlier itself. Which term is missing? Very good. For the continuous existence of species. So that is missing. Therefore, this is also wrong. Evolution. Change in the genetic composition of a population from generation to generation over a period of time. Is it just a period of time? No, right? Over a long period of time. So again, this is wrong. Development. Irreversible changes that occurs during the lifespan of an organism. That's the answer. So answer is what? 5. The feature that shows living beings as well as non-living beings is which character? 11th. What is the answer? Tell soon, tell soon. What is the answer for the 11th question? Nice. It's the growth. 12th question. Select the incorrect statement regarding characteristic of organisms. 12th. Give me the answer soon. Incorrect statement. They're asking about incorrect statement. And you all have to say me the reason why that's incorrect, Kayla. Okay, it's the third answer. Why is it wrong? Why is it wrong? Which term is missing? Very good. It should be what? Irritability and coordination, not just irritability. 13th one. What is the characteristic feature possessed by living organism? Like, what is the answer for this? Species, continuous existence of a species from generation to generation. Answer is 4. Nice. Statement is correct about water. Which statement is correct about water? Tell the answer soon. For the 14th question, which statement is correct about water? Very good. It's the second answer. Water function as a thermal buffer due to high specific heat. First one, see, water in solid form, is it for the bonds are fragile? No, right? It's the liquid form. Small insects on water can walk on the surface of the water due to cohesion, not adhesion. Only the cohesion between water molecules allow water to act as a transport medium. No, both the adhesion is also helping. Ice floats on on the surface of water bodies as water has a minimum density. Is it minimum density? No, right? It's the maximum density. What is the answer for the 15th question? What is the answer? Tell soon. 15th. Very good. 15th answer is 1. Growth and development are two consequent processes unique to living things. Passing genes that control specific physiological, morphological, and behavioral characters of organisms from generation to generation is called as what? Final question. Answer soon. Very good. It's the inheritance. Very good. Inheritance. So with that, unit one is done. I hope it's clear. Do you all have any doubts in unit one? Shall we move to the next part? Okay. So let's go to the next one. The cell crash. Yeah, we did this one. And elemental composition, we have major elements which are making up the 96 percentage of living matter. And then the remaining four are, for this, remember like this, Ka, P, K, S. That's how I remembered. Ka, P, K, S. These are the remaining main four elements. And then we have the trace elements, sodium, chlorine, magnesium, all these things. Remember them as well. And there are 92 elements on earth, and only 20 to 25 percentage are essential for healthy life and reproduction. And 25 elements, not, see here, there is an error. So 25 elements is essential for humans and 17 elements are essential for plants. We did these characters of water and carbohydrates. So the carbohydrate has a monomer called monosaccharide. This is the general formula, and disaccharides, polysaccharides are there. So in this, for this unit, let's go for the question itself directly because this is a big unit. No? So we'll go to the question itself, and from questions we'll come to the theory and refer the theory. Okay, I want you all to try the first 10 MCQs soon. Try the first 10 MCQs. Like first, this page, y'all can try the first 11 MCQs. Try the whole page soon. Okay. I'll give y'all 10 questions. Now 6:25. Finish this before 6:30. Just 5 minutes. Okay, let's go for the first question. Which of the following is not a physical or chemical property of water? Tell me the answer soon. Not a physical or chemical property. What would be the answer? Soon, soon. Have to be very active. You all have only 15 days. First, see, charges are unevenly distributed within the water molecule. That's correct. Hydrogen bonds are important to maintain all the physical and chemical properties of water. That's correct as well. And then only when the water is in its liquid state, its hydrogen bonds become very stable. Is it stable? In the liquid state, is it stable? Is the hydrogen bond stable in the liquid state? No, right? They are fragile. So the answer is 3. Temperature of the water at the bottom of the reservoir in the polar region is 4 degrees Celsius. Water is a vital chemical constituent of living cells due to the versatility as a solvent. Okay. Second one, which of the following feature is correct regarding importance of physical property? Remember, they are asking about what property? Physical property of water for organisms. So what's the answer for the second question? For the second question, what's the answer? What is the answer for the second question? Very good. It's the third one. See here, high specific heat capacity, support cooling of the body surface of terrestrial animals. No, we did the same question. No, the same question is repeated here. Which one of the following can be hydrolyzed in human body? Again, the same question. Lactose. Which of the following statement regarding water is incorrect? Which statement is incorrect? See this question for fourth question. What is the answer for the fourth question? This is a nice question. The second statement is very nice. Very good. It's not 54 percentage. What is the percentage of water inside the living body? Percentage of the water inside the living body is it 70? No, not 70. What's the percentage of water inside a living body? Very good. It's the 60 percentage of water. So all the other statements are correct. Only this one is correct. Not 54 percentage. It has to be what? 60 percentage. Fifth question. Which of the following definition is most accurate? We did this thing as well. So the answer is what? Development. What is the property of water? Allowing it to act as a transport medium. Sixth question. What's the answer? To act as a transport medium. Who is helping? Very good. Both cohesion and addition. Nice. High heat of vaporization of water causes. High heat of vaporization of water is causing what character? What? High heat of vaporization. Very good. Releasing much heat energy with minimum loss of water. Releasing much heat energy with the minimum loss of water. So answer is 2. Which of the following doesn't show color change? We have Benedict test. Who is showing the color change for Benedict test? Tell me that first. Who is showing the color change for Benedict test? Very good. Reducing sugars. So which is a non-reducing sugar that doesn't show color change for Benedict? There is a non-reducing sugar, like the disaccharide especially. Which disaccharide doesn't show the color change for Benedict test? Nice. It's the sucrose. Very good. And also tell me what is the test we are doing for lipids? Lipids, what is the test we are doing? For lipids, Sudan 3. Very good. For proteins, very good. It's the biorate test. So those are the 3 tests. And why do we do the iodine test? Iodine test is done for whom? Done for which biomolecule? Very good. It's done for the starch. So here, which of the following doesn't show a color change with a Benedict reagent? Who is not showing the color with Benedict reagent? Tell the answer now. For the eighth question, what's the answer? For the eighth question, what is the answer? It's the glycine, because glycine is a what? It is a protein. Okay, glycine is a protein. So the answer is one. Which of the following can be the molecular formula of a polymer made from condensation of glucose? Let's see who is going to say the answer for this question. This is a tricky question. Ninth question. What is the answer for the ninth question? You all found out the answer, no? Yeah, that's the third answer. Okay, I'll explain all this one. Because here we are using what? We are using 10 monomers, okay? 10 monomers. So totally we have like the 10 monomers of glucose. So 60 carbon, 120 hydrogen, and 60 oxygen would be there initial. But after condensation, okay, after condensation, remember when you have 10 molecules, okay, 10 monomers, and if you condense those monomers, 9 water particles, like one less than the number of monomers would be removed. So if we have 10 monomers and if we condense them, 9 water particles would be removed. If we have 5 monomers and if we condense them, 4 water particles would be removed. So by condensation of the glucose, what is happening? Now 9 water particles are removed. Okay, 9 water particles are removed. So 9 into H2O, 9 into H2O. So how many hydrogens would be removed? How many hydrogens would be removed? Very good. 18 hydrogens would be removed. And oxygen, how many oxygen? 9 oxygens would be removed. See here, this is the initial one. C60, H120, O60. Now from oxygen, 9 are getting removed and it is becoming what? 51. And from hydrogen, 18 are getting removed and it is becoming 102. Did you all understand that? Understood? Understood, no? Very good. Select the incorrect statement regarding biomolecules. Incorrect statement. Which statement is incorrect? First one, no, phospholipids and cholesterols are biopolymers found in the components of the cell membrane. First statement is wrong. Okay, that is because why? Both the phospholipids and cholesterol are lipids. They are not biopolymers, or else they are not macromolecules. So they are not considered as either biopolymers because biopolymers are what? They are large molecules made up of repeating monomers. No, and like proteins, nucleic acids, polysaccharides, those are the biopolymers. And lipid is not a biopolymer. And yeah, next question. Which group contain only lipid molecule compounds? Which is containing only lipids? 11th question. Which is consist of only lipids? Only lipids. Which answer? What is the answer which consists only lipids? Very good. It's the third answer. Vex, cutin, and steroids. Okay, third answer. All the others are consisting of different substances. And now I want you all to try these questions. 12th question, 17, and then y'all can, this is easy. I'll try this as well, okay? Mm, and after that, y'all can try this one, 15. This question. Try the questions which I put a tick mark, okay? And if we have time, we'll do the remaining questions. So for now, I'll give you all the important questions. Yeah, do all these questions. 12, 13, 15, 16, 17, and then 21, 23, 25, 26, 28, 29, 32. Do all those questions. I'll anyways put those the numbers of questions in the chat box. Now it's 41. Do them before 50. 6:50. Try to do them before 6:50. And if you all have any doubts in between, ask. Let's start the discussion. So, 12th question. Three test tubes are prepared as follows. So, 1 hour later, Benedict solution was added to all three. So, which will give the red precipitate? It means which will react with the Benedict. So, what's the answer? What is the answer? What's the answer? Okay, let's see. Adding 2 centimeter of glucose solution, obviously it would react, right? Adding 2 centimeter of sucrose solution with dilute HCl. When we added dilute HCl, what is happening? The sucrose would be broken down into glucose plus fructose. Both the glucose and fructose can react to Benedict. Adding 2 centimeter cube of starch solution with amylase. Starch with amylase. What is the substance it is giving? Starch with amylase. What will it give? Starch with amylase. I renamed myself as the chat here, so please drop the chats there. Okay. Maltose. So again, maltose can react. So the answer substances that maintains the fluid of the plasma membrane. Who is maintaining the fluidity of the plasma membrane? Very good. Phospholipids and cholesterol. So answer is A and B. Nice. 15th one, which one of the following is not a biological polymer? Who is not a biological polymer? Answer for the 15th question. Very good. It's the ATP. Nice. Which of the following statement is correct? What is the answer for 16th question? 16th. 16th. 16th question. What is the answer? Thirteenth, the answer is 3. What is the answer for 16th question? For the 16th one, what could be the answer? It's the first one. See here, when dilute H2SO4 and Benedict solution is added to a solution, sucrose and heated, it would finally result in what? Brick red. Because sucrose would be turned into, by adding the acid, it can be turned into glucose plus fructose, and glucose plus fructose will react with the Benedict, and finally they will give brick red precipitate. When alkaline copper sulfate is added to egg white, it gives a red color. That's not a possible thing. When I2Ki is added to a solution of corn flour, the brownish yellow color appears. Corn flour is consists of starch, so it doesn't give brownish color yellow. It is giving what? Purplish blue or else dark blue. Sesame oil appears yellow in color when Sudan 3 was added. What is the color the lipid substances will give? What is the color of the lipid substances when added this thing? What is that? Sudan 3. When we add Sudan 3, what is the color of the lipids? Very good. It is giving red globules. Okay, it is giving red globules. When Benedict solution is added to a solution of galactose, a red precipitate appears. No, right? It doesn't react for the galactose because galactose is what? Galactose is a type of polysaccharide, non-reducing sugar. 17th, which of the following is incorrectly indicating the test and observation with the reasons for the observation? What is the answer for 17th? 17th, what is the answer? Very good. It's the fifth answer. Iodine test, yellow brown color, absence of starch. I said this already. Okay. No, no, wait. Iodine test, presence of starch. This is correct. Byrate test, purple color, presence of proteins. Benedict test, brick red color, presence of reducing sugar. Sudan 3, red fat globules, presence of iodine test, yellow brown color, absence of starch. That's also correct, no? The last statement, see that? In the absence of starch, it will give the same color, no, like as it is. No arguments in that? What do you think? Fifth is correct, no? So there is no any answers here. No answer. We have to give all for 17th question. Yeah, free marks. But don't expect this in A-level. Okay? This has to be all. And then go for the 21st question. Which of the following is a polymer containing glycosidic bond? Who is containing glycosidic bond? Tell me this soon. Uh, what is the bond present in this one, polysaccharides or disaccharide? The bond present in polysaccharides or disaccharide. What's the, yeah, it's a good short note. What is the bond present in very good, glycosidic bond, which is having the, what is the bond in proteins? Proteins, what is the bond? Proteins. Is it ester bonds? Proteins? That's for lipids, ne? Ester bonds? Oh, am I wrong? Very good. It's the peptide bonds. Okay, peptide bonds is for proteins. What is the bond present in nucleic acids? Nucleic acids, what is the bond? Phosphodiester. The bonds in lipids is ester bonds. Okay, these are the four bonds. So here, glycosidic bond is present in which substance? It is present in the pectin, right? Pectin is a polysaccharide or monosaccharide or disaccharide. Pectin, what is pectin? Pectin, very good. It's a polysaccharide. Go for the 21st question, sorry, 23rd question. Select the correct option containing correctly matched organic compounds found in organisms, their building units, functions respectively. So what is the correct one? 23rd. What's the answer? I want everyone to respond. Don't be silent. What's the answer for the 23rd question? 23. Very good. Third answer. Immunoglobulin is an amino acid and also it is involving in neutralizing the foreign substances. Hemicellulose. Hemicellulose consists of pentose and hexose, but it is not the organic compound, right? This is the monomer. Okay, this is the monomer of hemicellulose. Preventing desiccation. What is the function of hemicellulose? Function of hemicellulose. Tell soon. See here. Here you all can go through this chart for the polysaccharide summary and then for the functions of carbohydrates. And after that, for the proteins, you can have this thing. Okay, proteins are here. Oh, that chart was not added. See here, that's why I say be with the resource book as well. The chart was not missed here. And then we have the, yeah, that's it. Yeah. And we have the lipids. So we have fats, phospholipids, and the functions of lipids are there. Proteins thing is not added. No, I have to add that also. Okay. So yeah, back to the question. So the function of the hemicellulose is what? What is the function of hemicellulose? Tell soon. Formation of cell wall component. In chlorophytes or plants or in both? Till that point will you? No. Hemicellulose is on only in plants. Okay, the cellulose is the one which is present on both chlorophytes and plants. So that point is also an important thing. Phospholipids, glycerol and fatty acids maintain the fluidity of the plasma membrane. Phospholipids, does it have the glycerol and fatty acids as the monomer? These are the monomer of lipids, right? Lipids only have the monomer as glycerol and fatty acids. RNA, ribonucleotides. RNA ribonucleotides, that's correct. Transmission of information from generation to generation. Is it the function of RNA? No, right? It's the function of DNA. DNA ribonucleotides, sorry, deoxyribonucleotides, providing site for polypeptide synthesis. No. What is the, whose function is this? Providing support for polypeptide synthesis. Whose function is this? Very good. I'm seeing only 5 people randomly giving the answers. Others, why are not active? It's the function of rRNA. Incorrect statement regarding enzyme. Incorrect statement regarding enzymes. What is the incorrect statement? Others, I want the response from y'all. 4, 2. What else? What is the other options y'all have? Huh? Incorrect, incorrect. See, the reaction catalyzed by each enzyme is very specific. Specificity of an enzyme results from the shape of the enzyme. That is also correct. The shape of the active site is not always fully complementary to substrate. Why? Because the enzymes are not rigid structures. The enzymes will start its catalytic activity after the enzyme substrate complex is formed. That is wrong because like during the induced fit mechanism itself, it can start its catalytic activity. Okay, so this is the answer. Some cofactors tightly bind to the enzyme and remain permanently attached. That's also correct. What is cofactors? What is cofactors? Tell the answer for that. What is a cofactor? What is a cofactor? Non-proteinous components which are essential for the catalytic activity of certain enzymes. Okay, at the point, certain enzymes, they are not just the enzymes, there are certain enzymes just have, like, not, it's not present in all the enzymes, it's present only on certain enzymes. 26th question, what is the answer? Yeah, nice. Correct regarding enzymes. Very good. Everyone are giving the correct answer now. So toxins are binding to the enzymes irreversibly. Who else can bind through irreversible bonds? Like irreversibly. Who can bind through irreversible, through covalent bonds? Very good. Toxins and poisons. Remember that irreversible. Whenever you get the irreversible binding the stuff, remember they are binding through what bonds? They are binding through the covalent bonds. And if they bind through hydrogen bonds or other weak chemical bonds, they are reversible. Okay. Activators affect the function of enzyme by binding to the active site through the. Does the activators bind to the active site? Can the activators bind to the active site? Very good. They are binding to the allosteric site. Allosteric activators will bind to allosteric site, but usually, like when they given as activators, they are binding to the regulatory site. Okay. And are they binding through covalent bonds? Are they bind through the covalent bonds? No. Then by what bonds they are binding? They are binding reversibly, right? They are not irreversible, so weak interactions. So that's how you have to analyze each and every questions. Like after the session, you have to go through the whole MCQ once again, okay? Then only you can be perfect, inshallah.
Bonds involved in the formation of enzyme substrate complex change due to higher temperature. It is happening in all the enzymes? No, right? Not on all the enzymes. So they haven't given specifically. Bonds involved in the formation of enzyme substrate complex change due to high temperature. This is correct, but they haven't mentioned this properly. Okay, like because they are like not all the enzymes are sensitive, no. Because only most of the enzymes are sensitive. See here, see here. Enzymes. Where is the enzymes? Mm, yeah, most are globular proteins and most of them are heat liables. Okay, most of them only heat liables. Not all these enzymes are heat liables, so that statement cannot be taken. Many competitive inhibitors bind to the active site of the enzyme irreversibly and change their shape. Competitive, nah, anyway, it should be what? Non-competitive only. Competitive inhibitors can bind to the active site, okay? But they do not change their shape. They are competing with the whom? Competing with the substrate, okay? Non-competitive inhibitors are the ones who are changing the shape. Coenzymes are proteinous, anyway. It should be what? Non-proteinous. 28th question. Okay, 28. Which of the following statements about enzyme activator and inhibitor is correct? What is the answer for 28? Very good. Some have given the answer as well. What is the answer for the 28? Okay, they bind to enzyme with weak chemical bonds. About inhibitors and activators bind to the enzyme with weak chemical bonds. ATP act as activator during irreversible reaction. Your answer is what? They are saying as the answer is 5. Allosteric regulatory molecules act as non-competitive reversible inhibitors. That's correct. Non-competitive inhibitors bind with enzyme at their active site, not binding to the active site. Um Antibiotics bind to enzyme of microorganisms permanently. They are binding temporarily. Okay. And then ATP act as activator. Is the ATP acting as activator or inhibitor? Tell that. ATP and ADP. Who is acting as an activator and who is acting as an inhibitor? Inhibitor is ATP and activator is ADP. Okay. So that statement is wrong. They bind to the enzyme with weak chemical bonds. Is this correct? Enzyme activators and inhibitors. First statement. Is it correct? 28. First statement. What do you all think about that one? 28 first statement. Why it's not correct? If it's not correct, why? If it's correct, why? I want the reason. Why it's not correct? Very good. Only it can be bind permanently as well. Very good. It can bind to but only most are reversible. Therefore, that statement is wrong. Nice. 29th question. Now, this is a good question. What is the answer you all got for 29? For this, you have to use a general knowledge pot duck. So, what's the answer you all got for 29? 29. What's the answer you all got? Oh, you all didn't try that question. 29? Why everybody are silent? Did you all got the answer or no? Nice. The answer is 5. Let's see why. Why here? This is the one explaining the allosteric regulation. Okay, see here we have the B. So this B can be oscillating between the two shapes. So it is either becoming active or else inactive. So what happens is this by X is there no, this X is coming and binding to this side. This is which side? This is the allosteric side. So when it comes and bind to the allosteric side, what is happening? It is activating the complex. And if it goes and bind to the inactive side, it will inhibit the reaction. So this Y is inhibiting and X is what? X is stimulating the reaction. Okay, so Y function as a non-competitive inhibitor. Can we take this statement? Y functioning as a non-competitive inhibitor? Is it acting as a non-competitive or competitive inhibitor? We can't say, right? We can't say because non-competitive inhibitor go bind to the enzyme, okay, and change the shape. It doesn't change the shape, no. So why cannot act as an non-competitive inhibitor? For the non-competitive inhibitor, it should go and change. Like if we have the enzyme like this, okay, this is the active site, let's say, and if the inhibitor goes and bind to this point, like any point except the active site, and if it changes the shape of this thing, okay, if it changes the shape of the active site only, we call that as non-competitive inhibitor. But this is not a non-competitive inhibitor. Second statement, Y forms covalent bond with enzymes. Are these forming covalent bonds? No, right? They are forming weak bonds because they are not attaching permanently. Y X act as ATP and Y act as ADP. Or is it the other way around? What can be? X is acting as a activator, therefore it should be the ATP, and Y is acting as an inhibitor. It is acting as what? It is acting as a ADP. So ATP. So X is ADP and Y is ATP. Y would increase the rate of catalytic activity by involving in cooperativity. Will it increase the catalytic activity? No, right? It would decrease the catalytic activity. Therefore, this statement is wrong again. B oscillates between two different shapes. That's correct. So answer is 5. 32nd question. Select the correct statement regarding enzymes. So what is the answer for 32? Did you all understand or you all have any doubts? 32. Correct statement regarding enzymes. Enzymes change the function of end products. That's wrong. Enzymes activate the metabolic reaction only within living cells. No, not only within living cells. Substrate molecules are larger than enzymes. Are the substrate molecules larger than enzymes? Yeah. Sorry, no. Substrate molecules are smaller than enzymes because they only have to come and bind to the enzyme. Allosteric molecules bind to the enzyme with non-covalent interactions that correct all amino acids which make enzymes are participate in biochemical reaction. Not all the enzymes are making the enzyme, but not all the amino acids are involving in the formation of enzymes. Only very few are forming the enzyme. So we can't say that. So the answer is what? Answer is 4. Is it clear up to this? Or you all have any doubts? Now you all can try out these questions. Okay, the questions I mark. I'll give you all around 15 questions to do. Try them out, okay? This is a good question. Yeah, try out these questions from here. 35. I put in the chat box what are the questions you are supposed to do. Okay. 35 and then do 36. After that, you all can try the 40th question. Then slowly go for 42, 44, and then you all can try 46, 47, 48, 49, and then 50, and next go for 52, 53, and 52, 53, 55, and then you all can try 56, 58, 60, 62, 63, and 65, 63, and then 65. Do all these questions I sent in the chat box. Do all the questions I sent. Now it's 7:17. I'll give you all till 7:30. Try to finish them. Okay, now let's go for the next set of questions. Okay, see the 35th question. What is the answer for the 35th question? Select the correct statement regarding an enzyme that shows allosteric regulation. What is the answer? Very good. Fifth statement, right? Regulatory molecules stimulate or inhibit the activity of the enzyme. Select the correct response regarding the nucleic acids. Okay, regarding nucleic acids, you have to identify the correct statement regarding nucleic acid. What is the answer you all got for 36? See the first statement, nucleic acids are composed of a backbone with repeating pattern of sugar phosphate units. Correct. They are linear polymers with nucleotides. Only thymine and cytosines are found inside the nucleic acids as pyrimidines. Who are the pyrimidines we have? What are the pyrimidines we have? Pyrimidines. Very good. Thymine, cytosine, and uracil. And then the other two are purines. So the statement is wrong. Always two antiparallel polynucleotide chains are found, not always. So again, that is wrong. Always the number of adenine bases is similar to the number of thymine bases in nucleic acids. This can happen, but not always. Okay, there might be mutations and stuff. No, so the answer is what? A and B. This is the main thing. Like this is the base pairing thing happening. But since it doesn't occurs always due to mutation and stuff, we can't take that one. Now go for the 40th question. Certain double-stranded DNA molecules consist of 1,300 turns. One turn consists of how many base pairs? One turn consists of how many base pairs? 40, right? Sorry, 10, right? 10 base pairs are there. And so how many bases will it have? So 1,300 into 10, and it will consist of what? Two bases, okay? Two bases. So 2,600 and have to be multiplied by what? 10. So totally we have how many bases? We have 26,000 bases. And if there is 26,000 bases, we have 25,999. What? This thing is there. Phosphodiester bond is there. We have to just what minus only one from the number of bases. Okay, so 99 2000 25,998 bases would be there. Okay, so we will have 25,998 because we have the phosphodiester bonds in between two phosphate. No, see here now we have two phosphate, this one nucleic acids. So in between both of these only we will have one. So we have to subtract one. So on either side we are having like that. No. So if we have four, we have how many bonds? We have two bonds. So that's what we subtract two. So if we have three molecules, see here we have how many bonds? We have totally we have six bases, but we have four bonds. That's how we go for the number of phosphodiester bonds. So the number of phosphodiester bonds is what? 25,998. And also 35 percentage is guanine. Therefore, what is happening? 35 percentage guanine means we have 9,100 guanine. And then A and T would be what? 15 percentage. So 15 percentage it means 3,900. So again 3,900 T would be there. So what is the answer for the 40th one? Answer is 5. No, the number of phosphodiester bonds of the molecule is less than 13,000. That is wrong because it is greater than 13,000. For the second question, number of nitrogenous bases contained in the DNA molecule extracted from mitochondria is 40,000. So this is the number of what DNA bases, which of which there are 18,000 adenine bases. Select the false statement about the DNA molecule. You have to select the false statement about the DNA molecule. So yeah, what is the false statement? 42nd, what is the answer? Has a double helix structure. The DNA will have the double helix structure. No, it would have the double helix structure, but that is correct. The number of purine bases exceeds the number of pyrimidines. Is it exceeding the number of pyrimidines? No, right? It should be equal. Contains 39,998 phosphodiester bonds. Is it correct? 42nd. Now answer that question. 42nd. Is it correct? The third statement. Is the third statement correct? Contain 39,998 phosphodiester bonds. Yes, that's correct, right? That's correct. We have two answers. The number of hydrogen bonds contained in the DNA molecule is 42,000. Is it 42,000 number of hydrogen bonds? How do you calculate? How many hydrogen bonds are present between A and T? Between A and T? You have three bonds, and between A and T you have two or three. Nice. You have two bonds between G and C. You have three bonds. So how many adenine you have? You have how many adenines? How many adenines you have? Eighteen 18,000 adenines are there. Therefore, we have 36,000 hydrogen bonds between A and T. And then the remaining is what among the 40,000 ones, we have the G and C. The remaining would be what? G and C. So 2,000 G would be there. 2,000 into 3 means 6,000. So 6,000 plus 36,000. We have how many bonds? We have 42 bonds. So that statement is also correct. They are asking about the incorrect one, no? Okay. Give me a second. Okay, so the statement is correct. There is only one oricide. Oricide, you will be studying this in Unit 7 also. So this can consist of only one oricide. So this false statement is what? Second one. Okay, now let's go for the 44. Percentage of ATP produced by substrate level phosphorylation during oxidation of glucose to carbon dioxide and water in an active cell. So in an active cell, how many ATP is produced? Total number of ATP in an active cell. What is the total number of ATP in an active cell? Nice, 32. By substrate level phosphorylation, how many ATP can we produce? How many ATPs are produced by substrate level phosphorylation? Okay, 4. So 4 over 32, 12.5. So we can take the 12, which is the nearest answer. So they are asking the approximate answer. Biological process consuming ATP. How do we call the biological process which consumes ATP? Active or passive? They are active. So ventilation of lungs is the process which consume ATP. Correct statement regarding ATP. So ATP, what is the correct statement regarding ATP? 47th question. Act as the energy carrier in all living organisms, used in the transmission of nerve impulses. Yeah, that's correct. And then act as an allosteric inhibitor to inhibit the production of more ATP via cooperativity, not in cooperativity, allosteric regulation. Energy needed for all living process is obtained directly from ATP. Yes. When hydrolyzed, each of the two terminal phosphate groups absorb Neve release 30.5 kilojoules per mole energy. So answer is A, B, D. And then go for the 48 question. Percentage of ATP produced during oxidative phosphorylation of one molecule of glucose undergoing aerobic respiration in an inactive cell. In an inactive cell, how many ATPs are produced? How many ATP is getting produced in an inactive cell? Very good, 30. By oxidative phosphorylation, how many ATPs are produced? Oxidative phosphorylation, very good, 26, because the other 4 is produced by substrate level phosphorylation. So 26 over 30 is 86.867. Therefore, we can take the answer 87.5 approximately. Correct statement regarding cellular respiration. So, what is the correct statement for the 49th? The first step of cellular respiration is pyruvate converted into acetyl coenzyme A. No, right? It's not the first step. Okay. That's not the first step. Therefore, first statement is wrong. And then, second one, the in oxidation phosphorylation, the number of ATP produced is 30. Is it 30 oxidative phosphorylation? No, right? It should be what? 26. The final hydrogen except in ethyl alcohol fermentation is pyruvate. No, it is acetaldehyde. Energy compared to ATP is released in citric acid cycle. Energy compared to 18 ATPs. Is it 18 ATPs? No, right? 28 ATPs. From substrate level phosphorylation, 2 ATPs are made from glucose molecules in Krebs cycle. So that's the answer. 50th, which of the following indicates the correct order of the main stages and their end products in aerobic respiration? What is the answer for 50? What is the answer you all got for 50? 50-50. Very good. It's the first answer. That's just theory. And then 52, total number of ATP molecule that result from NADH produced during entire aerobic respiration of a glucose molecules. So from how many NADH is produced? 10, right? So for each NADH we can get 2.5 ATP. So we are getting 25 ATP. Amount of DNA in a gamete of organism is 36.3.6 into 10 to the power minus 2 gram. Total amount of DNA in a somatic cell of the diploid organism at NR phase. They are asking about the NR phase. Now the amount of DNA, okay, the DNA is what? 3.6, okay, the amount of DNA is 3.6. Now first you have to remember the gamete is consist of N, okay. In haploid only it will consist of how much? 3.6. So in the somatic cell, okay, in the G1 phase of the somatic cell, okay, in the G1 phase of the somatic cell, it will be having what? It would be having the 2N. Okay, it would be diploid because diploid cells only can undergo the this thing. What is this? Meiosis. Ne? So 2N means it consists of how many DNA? 3.6 into 2. So it would consist of 7.2 into 10 to the power minus 12 gram. And then they are asking about what? They are asking after S phase, what is happening? Replication of DNA is taking place. So after S phase, the amount of the DNA would be what? 4n, because sister chromatids are also produced. So inside the sister chromatids also, we have two DNA, like inside one sister, each sister chromatid we have two. Like this is how the sister chromatids are. No? So this is how the sister chromatids are. So anaphase is like this. So in anaphase, we would be having sister chromatids attached to each other. So this will also have what? This will also have like this, no, have the sister chromatids. So inside each of the chromosome we have DNA. So we have four DNAs here. So 4n would be there in the metaphase. So and then for after metaphase, what is happening? We are going to the anaphase. When anaphase is coming, what is happening? Now this will be going to opposite side. This would be going to the opposite sides. So now we have the 4n, and after that these would be going to the anaphase. So in anaphase, before the cytokinins, okay, because after the telophase only, inside the whole cell the cell is getting divided into two cells, okay. After the telophase only, the cell would be divided into two cells, but they are asking about what? Inside a somatic cell, inside the whole cell. So during anaphase, this is taking this, even though they are getting divided into two, they are present inside one cell. So this will completely have, here there would be two DNA, and also here there would be two DNAs. So this will have how much? This will have 4n. 4n would be the number of DNAs. 4 into n. Here we are taking instead of n 3.6. So 4 into 3.6 is what? 14.4 into 10 to the power minus 12 gram. 55th question. What is the 55th answer? 55. In mitochondrial matrix, okay, in mitochondrial matrix, we are producing 2 by pyruvate. 2 NADH is produced by pyruvate oxidation and 6 by the Krebs cycle. So 8 NADH are produced in total. 8 into 2.5 is 20. 56, what is the answer? If you all have any doubts, ask. 56. What could be the answer for the 56th question? Correct about cellular respiration. B and D. See here, the total number of ATP molecules produced by oxidative phosphorylation in the Krebs cycle, the same thing is repeated again, 8, 8, 18. And then go for the D statement. Fatty acids can join to the citric acid cycle after converting into acetyl coenzyme A. And the glycerol will be joining to the G3P. Okay. So yeah, that's the answer for the 56, B and D, second answer. And then you all can go for the 60th question. Correct statement regarding transmission electron microscope. So correct statement regarding transmission electron microscope. What is the answer? 60th. Specimens are magnified by 5 into 10 to the power 5 times. Is it in the transmission one? Okay, no. Less electrons may get displayed in cellular structures with dense training. See here, for this you all can refer this chart. We have the chart for the microscope. Okay. See the microscope. Used to study the internal structure is TEM. Surface view is SEM. Electron beams of the electrons are passing through the specimen in TEM. In SEM, electrons are reflected on the surface of the specimen. Specimen preparation here in TEM very thin and stained with heavy metals. In SEM, they are coated with gold prior to observation. Image formation, this will reflect the patterns of the image through of the specimen, and here how many electrons, like it will show scatter many electrons. And also image detail, more electrons are displayed in regions where structures are densely stained in TEM, provide three-dimensional appearance in SEM. So from that question, you all can go for the answer. So for the 60th question, you all can get the answer as 3. Living specimens cannot be observed. And you have the 62nd question. Few advantages of microscope. What is the answer for 62? All got. Ability to observe non-living specimens, not an advantage. Ability to observe colorful images, not an advantage. It's a like not on all the enzymes. Like this is asking for electron microscope. No, light reflected by using glass lenses. So advantage of light microscope, right? They're asking about light microscope. Ability to observe the images directly. So these two are the examples, C and D. Okay, C and D. So that's it. 63, given below is an illustration of eukaryotic cell. Which statement is mismatching? So what's the answer for 63? What's the answer you all got? What is A? A is formed by the arrangement of 9 triplets of structures of D in circular manner. D is what? D is the microtubule. So A is formed by, 9 triplets, right? From microtubules only centrioles are formed. Structure C and E produce network of intracellular membranes. C and E. What is C and E? ER, right? Endoplasmic reticulum. Peroxisomes are produced by the vesicles produced by periphery structures of B. Peroxisomes, never, it should be what? Lysosomes. Structure F is not present in the cytoplasm of an erythrocyte. F. That's correct, right? F is what? Mitochondria, not present in the cytoplasm of erythrocyte. Erythrocyte means red blood cells. Red blood cells do not have mitochondria, and also it doesn't have nucleus. Structure D is important in the formation of spindles in cell division. Yeah, we are forming spindle using that. Function of subcellular structures shown in the diagram. So what is the function of the subcellular structures found in this diagram? This is the diagram of what? Diagram of the ER, right? Synthesis, lipids, synthesize glycoproteins, production of transport vesicles. And then we have the, this is correct, lysosomes cannot be produced. Autolysis causing cell death, this is also not a function because this is an ER, right? It can be SER, and also the rough endoplasmic reticulums are there. And then worn out organelles is also a function of lysosomes. And then electron transport is not a function of this. Therefore, the answer is 1. Is it clear up to this unit 2? Is it clear? Okay, I'll have classes no at 8 o'clock. Yeah. Okay, that's fine. So after this, these parts are like somewhat easy, the cell cycle stuff and all. Okay, you all can try these questions by your own, and there is like 136 questions, I guess. Okay, there are 138 questions. Okay, from this part, from unit 2. So now, I'll say about like important things. Now we have this graph. No, we have this photosynthetic graph. Remember, this photosynthetic graph, there are 3 main graphs. Y is the longest one, no, the one who have the highest hump. The B is also like this, no, it is the longest one. So chlorophyll B is represented by Y. And carotenoids are having what? Carotenoids are having 2 humps. Remember like that. Okay. Carotenoids are having 2 humps, and the one which is remaining is the Z. So the one having the longest hump is B, and the one had 2 hump is carotenoids, and the remaining one is Z. So that's how we go for this graph. So this graph is important, and any other important points? So you all can try out the 138 questions, the remaining questions in this tute for the unit 2. And yeah, that's it about the unit 2 stuff. And also go through these charts, especially the charts. Okay, the charts are very important. Like you all can refer everything. See here, the light microscope and electron microscope. TEMNSEM. After that, you have what prokaryotes and eukaryotes. Then subcellular structures, function, all those things are there. That's a common thing we get. And this cell cycle, mitosis, meiosis, and all. And also we have the benign tumor, malignant tumor chart. Then we have the cancer and goals. So go through all those charts. So that would be helpful. And see here C3 and C4 plants. So this was also in the resource book. So go through all these charts and get the maximum use of all these things. And I think we have this thing in this tute. You can just press and go for that part. I remember, as I remember, yeah. If you press this part, you can go to the specific thing. You can't use this in Drive, even if you press the table of topics. Okay, the table of contents also. If you want to press that part, you can go to those specific sections. So whenever you refer these tutes, just go through this once again and try those revision questions. Okay. So can you all try those questions like within today? Shall I send the scheme tomorrow or the day after? Can you try these questions today itself? All the questions? Tomorrow. Okay. So I'll send the schemes today. It's Sunday, no? So I'll send the schemes by tomorrow night then. Okay, tomorrow night you all have the time to discuss do these questions, no? So try out all the questions and yeah, I'll send the scheme, correct it accordingly. Before that, refer all the 4 units. Okay. So with before starting the unit 5 thing, you have to revise all the 4 units. So let's have a 5 minutes break. And during that break, you all can refer these charts. Evo dive, okay? You all go for these charts, okay? Go for these 2 charts. Okay, let's have 5 minutes, like, or else take the break, will you? Let's go for that later. Take the 5 minutes break. Come back, not 5, 7 minutes. Take 7 minutes, will you? Come back at 8:05. Come back at 8:05. We'll finish the session at 9, okay? Come back at 8:05.
Okay, now it's 8:05, so let's start. I don't think we would be able to do unit 4. I thought of doing just go like very fast, but can't do like that now. So we'll do until unit 3 today, and I'll give you another time to do unit 4. So the next session would be on unit 5. So yeah, we'll do the unit 4 on another day, like separately. So in EvoDive, what you can do is there are many charts added in this. Okay, so let's see the origin of Earth, maybe. Yeah, here origin of Earth is like the basic thing, and eons, eras, all these things are there now. You don't need to memorize all these stuffs under these eras. Okay, like Paleozoic, Mesozoic, Cenozoic, you don't need to memorize all these stuff. But anyways, just go through them because there might be an MCQ. We can't make sure about that. Okay, even though they said that there won't be any questions from these parts. Anyways, go through this part. Okay, so these eras things, and then you have the theories of all these scientists, Lamarck, Darwin Wallace theory, and Neo-Darwinism, and the process of natural selection. I remember this as OVA CONA, the first two letters. Okay, OVA CONA, overproduction, variation, competition, and survival of the fittest, and natural selection. So make some mnemonics to remember all those stuffs. And then we have all these things, and finally we are coming to the bacterial domain. So you can refer these images, like these images are from, like we use the AI to get these images, so there might be any errors. Okay, so don't just rely on these things, but go through the notes as well. And the cyanobacteria thing is also there. So go through the whole unit, whole tute, and y'all can try these questions, and especially these charts. Okay, this chart was in the resource book, but go through these charts. There are the charts. See here, we have the charts for Euglena, Paramecium, and Amoeba, and also for these three things, these four. This is very important. The chlorophyll component, and also then chlorophyll. This one cell wall composition. See here, cellulose. Here we have cellulose and alginic acids. And then we have what? Pectin and silica. After that, the thallus structure. This is also very important. Color green, greenish red, olive green, all those stuffs are there. So go through all the charts in Unit 3. Remember, to study Unit 3, you need many comparison charts. So if possible, if you're a 27 student, you all can make the comparison charts. Or if you're a 26, you all can refer these things. And this thing, remember the stored food and photosynthetic pigments. Okay, the stored food and photosynthetic pigments in everything, because usually they use the question from this part. So here A and B, here we everything would be having A. Here A D phycobiliproteins, here AC xanthophyll, here we have AC carotene and xanthophyll. So these are the photosynthetic pigments, remember them. And then when it comes to plants, again I added images, but don't depend on images. And here we have the chart. This is in the resource book as well. Again, we are going to the fungi. Fungi also I added a chart. Here, see. So this through this chart, you can refer all these parts. Remember the sexual and asexual reproduction thing, and also the nutrition wise. Separately study those parts. And eukarya also I added the this one chart separately for invertebrates and vertebrates. So in eukaryotes also refer these invertebrates and vertebrates chart. This is for the invertebrates. So you can refer this chart for invertebrates and the chart for the vertebrates. And also I have to say this thing. The characteristics of phylum chordata. Study this well, there might be a question this time. Okay, maybe a short answer or something. So characteristic of phylum chordata is a very important part. Okay. Then you all can go to this chart, the final chart here. Okay, this chart is also important. So through these charts, you can refer and answer the questions. Now let's go for the questions. So I'll randomly select some questions and give. Try the fifth question, 16th question, and then go for the 13th. And you all can try 21st, 20, and then try the 23rd one, 31, 38,
After that, you can go for 37 and 48, 43. Do until then. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Yeah, 11 questions are there. I'll give you all 10 minutes. Now it's 8:10. Try to finish by 8:20 these questions. 5, 16, 30. I'll put in the chat box. Do these questions soon. What I can say is now the time is very limited, no? So you all can't depend on anyone's, okay? You all can't depend on anyone's help. You all have to work by your own. So for this, what you can do is now you have to give the maximum time for yourself. Don't go to the theory classes now and waste your time. Try to work by your own. Like now, the self work is the most important thing. So do most like questions as much as you can. Don't do like if you are like in A level, it's okay. You can try full papers, but if you are even at C, S, or W, you can try like unit wise. Now we are doing like this. Now today we'll be done with the unit 1, 2, 3. So what you can do is you have to refer unit 1, 2, 3 before the next session, and you have to try all the things what I said earlier, this MCQ tute and the biocheck tute, and then after that you can go for the SASD. And you have to correct your own self because you can't expect anyone to do these things for you because everyone has helped you all these 2 years now. So now it's time for you. So you are the one who have to work right now, like sacrifice everything. Don't just say reasons and just skip these stuff. Make sure to do all these stuffs, okay? Within these 15 days, only 15 days. Now after that, you all can be free forever. If not, only the problem will come after that. You have to do the second try again. So yeah, do all the things now itself, like today itself, complete them. So you would be free for the next 2 years, like for the next part of your life, you would be free. If not, you have to do the same thing again and again. Okay, now let's move for the questions. Okay, see here, indicate the occurred just before diversification of large trees. So for this, you can refer the tute. No, you can go to the tute. Which part? Diversification of large trees. Mm, see here, we have the things. Large trees were diversified on which year? We have that, no? 1.8 billion year is there. In 1.8 billion, what is happening? Eukaryotic organisms are originated. And 1.2 billion years ago, oldest known protist, which is similar to the red algae, was origin. And before 700 million years ago, sponges were evolved. And then ancestors of arthropods and chordates were evolved 670 million years ago. And then after that only animals were depending on the algae or plants, and then the land was colonized by what? Fungi, plants and animals. So this colonization of land by fungi, plants and animals took place 500 million years ago. And plants possess vascular systems. After that, arthropods were the animals colonizing the land. And then tetrapods were, which are evolved from the free lobed fin fish, they started to colonize the land around 365 million years ago. And divergence of human lineages from the primates were took place 6 to 7 million years ago. And finally, the human species were origined 195,000 years ago. So these are the years you all have to remember. So go to this question now. Incidents which occurred just before diversification of large trees. So large trees diversification before the large trees. So what is the answer you all got? What happened before the diversification of large trees? I want the answer from everyone. What happened before the diversification from large trees? See here. We have the diversification of, there are different themes. Extensive forest of vascular plants were diversified. And then first seed plants were diversified. These are like diversification related to plants. Okay, here also plants were colonizing the earth. And then we have the diversification of vascular plants. After that, where do we have about the plants? You can go and highlight all these things. Cone bearing plants were coming in Mesozoic era. And then you have the gymnosperms. Okay, they are continued as dominant plants during Mesozoic era. And then flowering plants were evolved and they are diversified again in Mesozoic era. And then the dominance of angiosperms were increased during the Cenozoic era. So these are the things related to plants. So we have the first food webs on the earth appeared. These food webs are appeared just hours after the diversification of trees. Okay, because these food web what yeah food webs are appeared after the plants. Okay, because without plants we can't do no. So the fourth answer. Let's go for the fourth answer. Appearance of sponges, no, it is also took place before the plant. 700 appearance of sponges, that's the one. Okay, it took place before the what? Before these plant have evolved. Okay, 700 million years ago. Some state. Then let's go for the next question. Question number 13. Which of the following is incorrect statement about methane producing bacteria? Who is the methane producing bacteria? What is the methane producing bacteria? The methane producing one. Who is the methane producing bacteria? Methanococcus. Okay, Methanococcus. Methanococcus is the methane producing bacteria. What is the cell wall component of Methanococcus? Cell wall component of Methanococcus. What is the cell wall component of it? What is the cell wall component of? Is it proteins and polysaccharides or peptidoglycan? Very good. Proteins and Polysaccharides, right? It is the proteins and polysaccharides. Is it living in extreme environmental conditions? No, right? It's not living in the extreme environmental conditions. Some hydrocarbons in membranes lipids are branched. Some they have given us some can be branched. Their growth cannot be inhibited by antibiotics. That's right. What are the two antibiotics we had? The two antibiotics. Let's go here again. Okay. Two antibiotics. Two antibiotics. What are the two antibiotics we had? See here. This is the archaea. So we have the see here, methanogens. So methanogens are the ones they are speaking here. So methanogens of Methanococcus is an example. So they are living in more moderate environment, and we have like extreme environment organisms, but methanogens is not a organism which is living in extreme environment conditions. And then we have the characters. See here, unicellular prokaryotic. Like peptidoglycan, cell walls are made up of proteins and polysaccharides. Size is 0.5 to 5 micrometer. And the ones who live in the more moderate environment is methanogens. And then they can also live in anaerobic guts of cattle and termites and other herbivores. After that, let's go for the domain bacteria, domain three domain classification. So three domain classification in this see here characters of archaea. They are prokaryote proteins and polysaccharides. Some are branched, some are branched. Not everything are branched, some are branched. Histone proteins are present in some species. Again, some species. Circular chromosomes are present. Introns in genes are present in some species. And the initiator of the amino acid protein synthesis is what? Methionine. And growth is inhibited due to the what? Growth is not inhibited due to streptomycin and chloramphenicol. Okay, streptomycin and the chloramphenicol are the two antibiotics we have. So their growth is not inhibited. And then we have the growth at temperature above 100 degrees Celsius. Only some species can grow above the temperature of 100 degrees Celsius. And yeah, so see here, so all the other statements are wrong. So the answer is 2. So 2 never 3. Sorry, 2 is the correct, incorrect one. This is correct. And several RNAs are involved. That's also correct. That was also in that chart. Next, let's go for the 18th question. See here, a person who classified plants according to hierarchical levels of genus, order, and species, genus, order, and class. See here, now let's go for the classification thing. The scientist part. See, Aristotle. What did Aristotle classify? He is the one who classified organisms scientifically at the beginning first and divided them into what? Plants and animals. And then we have whom? The Theophrastus. Theophrastus is classifying what? Plants according to habitats and also according to lifespan. And then we have Carolus Linnaeus. He is introducing the binomial nomenclature and hierarchy orders is I remembered like SCOG. SGOC. Okay, SCOG. So species, genus, order and class. And two kingdoms, the kingdom Animalia and the kingdom Plantae. So these are the things introduced by the Carolus Linnaeus. After that, we have Ernest Haeckel. He is the one introducing the third kingdom Protista. Remember, see here, Protista phylum. So he is the one introducing things which start with P, Protista and phylum. And Robert H. Whitaker, he is the one giving the five kingdom classification: Monera, Protista, Fungi, Plantae, and Animalia. And also, this is mainly dependent on what? Cellular organization and mode of nutrition. Okay, this is mainly depending on the cellular organization and mode of nutrition. And Carl Woese is the one which is who is dividing organisms in according to three domain classification. We have bacteria, archaea, and eukarya in that. So that's it about the classification. And remember, present system of classification is mainly based on molecular biology and evolutionary relationships. Okay, these two things are the ones in which the present system of classification is based. And there are five main things which they considered during the present system of classification. These are the five main things: sequence of bases of DNA of important genes, okay, of the important genes, sequence of bases of DNA of mitochondria and chloroplast, and then base sequence of ribosomal RNA. After that, sequence of amino acids in common proteins, okay? Not in all the proteins, it's in the common proteins and molecular structure of the cellular components. So these five are the things that are considered during the present system of classification. This can be questioned in a structured essay question. Okay, so see here, who is classified this SGOC? Carolus Linnaeus. Okay, so the answer is one. Let's go for the 20th question. Which of the following response include organisms contain peptidoglycan in the cell wall? Peptidoglycan in the cell wall. Remember, for this I haven't included this in the this thing. What is that? The evodived, but this example was there. I'll take you all through the example. Wait, give me a second. Okay. See here in this chart, you have the examples for all the organisms. So for bacteria, we have cyanobacteria, nostoc, Anabaena, Escherichia coli, Salmonella typhi. And for Archaebacteria, we have Methanococcus thermococcus. Even though the Helobacterium has a similar name as of bacteria, this is a type of Archaebacteria. Remember that part. And also for this thing, eukarya, you have many examples. No need to say for that. So we have Anabaena and who is having peptidoglycan? Archaea or bacteria? Who is having peptidoglycan? Go for this tute again. Archaea or bacteria? See here, peptidoglycan is present only in the cell wall of the bacteria. Okay, only in the cell wall of the bacteria we have peptidoglycan. So the answer should consist of what? Peptidoglycan consisting of bacteria. So Anabaena and microcyst, are they having the peptidoglycan? Anabaena and microcyst. Tell soon. Anabaena is what? Very good. Anabaena and microcyst are two types of microorganisms, sorry, bacteria. So they can consist of peptidoglycan. Cyanobacteria is also coming under bacteria. No? Lingvia and Helobacterium. We can't take Helobacterium. Again, we are having Thermococcus. And then we have the Saccharomyces. Saccharomyces cannot be taken. And Saccharomyces is a yeast. Clostridium, Methanococcus, this cannot be taken. So the answer is 1 again. 21st. Classification made based on the natural classification. Who classified organisms based on natural classification? Very good, Carlos. See here in this thing, you can go to that part. That was also not in the thing I have made. See here, binomial nomenclature before that. We have the scientist, no? So in this paragraph, okay, remember natural system with interpreting evolution relationship. Okay, after this only, okay, after this only, Carl Wood classified the organisms. Okay, so this paragraph is important. So after the Darwin's theory of natural selection and all, and the unitary origin of life, Carl was classified organisms based on this. So Darwin's theory of evolution and unitary origin of life, taxonomists began to use the natural system. So natural system was used by whom? By the scientist Carl Woos. Okay, so it is the Carl Woos. Then go for this question. Select the common characteristic of Euglena, Paramecium, and amoeba. Now we have to go for the chart. See here. Euglena, Paramecium, and amoeba. So all of these are living in fresh water, but only the Paramecium is confined only to the fresh water. Even though Euglena and amoeba can living in marine, Paramecium is living only in the fresh water. And then cell type, all of these are unicellular. Cell wall, Euglena and Paramecium doesn't have, three of them don't have cell wall, but Euglena and Paramecium have what? They have pellicle. Okay, both of them are having pellicles. And the shape, these have definite shape. Euglena and Paramecium are having certain shape, but amoeba do not have a shape. Locomotion, Euglena is using flagella, Paramecium is using cilia, and the amoeba is using what? Pseudopodia. Nutrition, euglena is mixotroph, can do both autotrophic and heterotrophic, can produce its own food as well, but the other two are heterotrophic. Chloroplast is present because it's an autotroph, the other two absent. And one nucleus is present in euglena and one nucleus is present in amoeba, but the paramecium is having how many nucleus? Two types of nucleus are there. One is the meganucleus and the other one is what nucleus? Micronucleus. Okay. And the food intake, so absorb or photosynthesis, they are using paramecium use oral grooves to engulf the food, and the pseudopodia is used by the amoeba to engulf the food. Vacuoles, contractile vacuoles are present in all three, but the euglena is lacking the food vacuoles, the other two are having food vacuoles. And these are special features, eye spots are present, pockets at one end to emerge the flagella. These are special characters of what? Special characteristics of the euglena. And the cilia for locomotion and oral groove is a special character of the paramecium, and pseudopodia is a special character of what? Amoeba. Now let's go for the question. Cytoskeleton hold fast genes with introns. Are these things present in euglena, paramecium, and amoeba? No, right? These are not present. Okay, these are not present. AUG start codon, many forms RNA polymerase, sexual reproduction. No, sexual reproduction cannot happen. And cell wall 9+2 structure, contrail vacuoles, this cannot occur. But they have ribosomes, sorry, they have iterase ribosomes, but not everyone are having pellicle. Genes with introns, obviously, because they are what? They are eukaryotes, so they have genes with introns. AUG start codon, it is the methionine, and then they have microtubules. So answer is 5. Okay. Now let's go for the 20. Which question? 31. Before that, let's go for a fungi question. Some or else plant question. Phylum of organisms evolved first during plant evolution. Who is evolved during plant evolution? First evolved during plant evolution. Who is evolved? For this thing, you have to refer this chart. Okay. That was also not included. Okay. Wait, I'll show you that graph. This graph is there. Now, this graph is very important. See here, they have okay. They have given the chart for this. See here now, who is formed first? Liverworts. Liverworts are the ones who are formed around 500 million years ago. And then only who is coming? The mosses are formed. So mosses is like around 475, let's say, or else 450 or 50, 55 again. And then we have the hornworts. Hornworts are formed around maybe 440. And then we are having what? Lycophytes. Lycophytes are formed around maybe 425. So the one who formed first is liverworts and the one who formed last is angiosperms. This chart, most of the students, they used to ignore this chart. So study this chart well because there might be a question. So from this chart only you have to answer this one. So the phylum Hepatophyta is the one which is evolved at the beginning. Okay, phylum Hepatophyta. Why it's not working? Phylum Hepatophyta. And then let's go for any other fungi questions are there? Protist chlorophyll stuff is there. No, I said that also, right? Let's try to revise and go. See here, I said this chart, so from that only you have to answer this question. True regarding the protist. So all protists contain chlorophyll A. No, right? All of them doesn't contain chlorophyll A because only certain algae would be consisting of chlorophyll A. And then all of them are unicellular. Are all of them unicellular? No, right? Not all of them are unicellular. Some multicellulars are there. All protists possess cellulose cell wall. No, some have L-genic and stuff. All protists are eukaryote. That's correct, right? Protists are eukaryotes. And then we have what? All protists are photosynthetic. Not all the protists. Some protists are photosynthetic. So answer is 4. And let's go for a question about amphibians. Yeah, we have amphibians. So yeah, let's go for amphibians. I'll just refer the note for the particular section and go. So it would be easy now. Amphibia is an invertebrate. We have the amphibians here. No, amphibian is a vertebrate. Sorry. Amphibian is a vertebrate. So these are the characters of amphibia. Amphibia, especially remember this frog one. So example is toad, frog, and lithiopis. Lithiopis is something special because this is the one which can do internal fertilization. All the others are doing external fertilization. So this guy is doing the internal fertilization, and this is having a. But see here, this is a snake-like structure. Okay, this is having a snake. This is a snake and has like three yellow color lines on top of the body. I guess that's the structure, and this is the first animal to invade the land, but they need water to complete their life cycle. And found only on land and freshwater, no marine species. This is a special character of amphibians, no any marine species. Skeleton composed of bones. This is a common character for all the vertebrates. First species to possess the limbs. This is the first one who is having the limbs. And then tetrapods with digits. Larval stages respire through. The larval stages are respiring through what? Through the gills. And some can respire through the skin and buccal cavity, or else it can also exchange the gas through lungs. So there are four organs to do the gas exchange: gills, skin, buccal cavity, and lungs. And they are what? They are ectothermic in the sense cold-blooded. Thin, moist skin with glands. Okay, they are having glands but no scales. Reptiles do not have glands. They have what? Scales. That's the difference between reptiles and amphibians. And sensitive to environmental changes. They have nictitating membrane. See here, this is the nictitating membrane. Like there would be something sliding on top of their eyes. That is called as what? Nictitating membrane. And also it has what? Tympanic membrane. See here, there is a tympanic membrane. See where my cursor goes. Okay, so this is the tympanic membrane. Also it has what? Nictitating membrane. Most are doing what? Fertilization, external fertilization. That's why they have given most, because Ethiopis can do internal fertilization and eggs do not have any shells. So these are the characters of amphibians. So they are endothermic animals. This is wrong. And then young and adult forms have excretory products, different excretory products. Yeah, we'll be studying this in unit 5. So then the third statement, you can have the third statement. Adults have ventral heart. Obviously, ventral heart is a character of chordates. Let's go there. Oh my God. Let's go for the characters of chordates. Let's revise that also. Chordates. This is the characters of chordates, okay? See here, there is a notochord, longitudinal, flexible rod between digestive tube and nerve cord. This is the notochord, okay? This thing. Ayyo. Okay, this is the notochord which is going through. The red color one is the notochord. Okay, this is going between what? Between the nerve cord and the digestive tube. The tube, the light pink one. Wait, show. Okay, this is the digestive tube. Okay, this is the digestive tube, and this is the nerve cord. And this is the red color one is the notochord. And then we have dorsal hollow single nerve cord. The nerve cord is the yellow color one. And then we have what? Pharyngeal slits are there. Okay, these are the pharyngeal slits. Okay, these are the pharyngeal slits. After that, we have what? Muscular tail. See here, this is the muscular tail. And then we have the ventral muscular heart. The ventral muscular heart is also present inside this what? This chordate. So these are the 5 main characters of the chordates. Phylum Chordata. Okay, I construct. Yallah. Okay. So we are in the 32nd question. So the adults have ventral heart. This is the answer. They lay shelled eggs. That's not a correct answer because amphibians do not have shells. Only young forms require water to complete their life cycle. Never everyone require water to complete their life cycle. So answer is 3. Then let's go for yeah. Trimerous flower part. So 37, for this you are supposed to go through this chart. The plants chart was there, no? Monocot and dicot chart. Uh, okay. Prefer the monocot and dicot chart. See here, monocots have one cotyledon, dicots have two cotyledon, and the monocots have fibrous root system. Yeah, see here, monocots are having fibrous root system. Embryo is having what? One cotyledon. Flower structure, they have what? They have the calyx and corolla not differentiated. And then pollen is having only one opening or one aperture. Stem anatomy, what has happened here in monocots? They are like scattered, and then we have the examples as grass, coconut, and rice. And also leaf venation, parallel veins are present. Pollens, only I said about pollens, no? Only one apical. See the dicotyledonous plant, here the dicots, okay? In this side we are having the dicots. So what is happened to dicots? Embryos have two cotyledons. See here there are two cotyledons. And also the root system is taproot system. Leaf venation, reticulate venation. Flower is pentamerous or tetramerous. And then stem anatomy, they have like visible ring-like structure. And also the calyx and corolla can be differentiated. So these are the characters of these monocots and dicots. So according to that, you can answer the 37th question. So for the 37th question, you will get the answer. Yeah, for the 37th question, you can get the answer as two. Second one is the answer. Okay, we have the second one is the answer. And let's go for the fourth 38. Correct statement regarding kingdom plant. So kingdom plant reduction in the gametophyte is a trend seen in the evolution of plants. So when going through from the bryophytes to the angiosperm, what happened is this thing was there. No, what is that? We had the gametophyte and sporophyte. No, so gametophyte was reduced. Okay, gametophyte was reduced when going from the bryophytes, so from non-vascular plants to vascular plants. So this statement is correct. Root tissues of living vascular plants resemble the stem tissues of early vascular plants. Is it correct? That was an exact statement in the resource. Okay, so that's also correct. Ancestors of members of kingdom Plantae had key traits of land plants. No, they had the key traits of what? Algae, right? The green algae. Members of kingdom Plantae evolved from a group of olive green colored protists. They are evolved from a green color protist. Liverworts are evolutionarily closer to hornworts than mosses. It's not the liverworts which are closer to hornworts than mosses. See here. For liverworts and mosses, we have a large gap like this. But for the hornworts and mosses, we have what? A very small gap. So hornworts and mosses are more closer than the liverworts. So answer is what? A and B. Third answer. And then 43rd question, characters of codeates are seen here. Now let's go for the codate characters. See here we have the codeates characters. Let's go over the final chart we had. Okay, these are the characters of codeates. So here we have different types of these things. Codeates. Now habitat, all of these are like aquatic creatures. You all can identify that no. And endothermic is only the aves and the mammals. Okay, aves and the mammals are the ones which are endothermic. And the skeleton mainly the cotton-rich thighs are having cartilage. All the others are having bones. Okay. And respiration, conic thighs can do by gills. There is no operculum against gills with operculum. And then we have the gills again for amphibians. Lungs are present for reptiles. And the other three things. So lungs are starting from amphibians, and body covering placoid scales for chondrichthyes, cnitoid and cycloid for ostrichthyes, and moist skin with glands for amphibians. And keratinized scales are for reptiles. Feathers are located present in legs, have scale in legs of the aves, they have scales, and hairs are having mammals are having hairs. So remember, the mammals and also the aves and the reptiles, all these have what? Keratinized things, because the scales of reptiles are keratinized, the scales in the aves are keratinized, and the hairs of the mammals are keratinized. So keratinized structures are present in reptiles, aves, and mammals. And then we have the heterocercal fin in the chondrichthyes and homocercal in the ostrichthyes. Also, we have a special structure called cloaca in the this one chondrichthyes, swim bladder in the ostrichthyes, nictitating membrane, tympanic membrane for amphibians, calcareous eggs for reptiles, beak for aves, and the mammary glands and diaphragm for mammals. And fertilization, internal fertilization is carried by chondrichthyes. Okay, ostrichthyes can carry out internal fertilization as well, but most of them are external. Also, reptiles, and then we have the aves, and then finally we have the mammals. They are also what? Internal fertilization. So you all have to study this separately. And also, oviparous means what? Oviparous means what? Putting eggs, okay, and viviparous means laying the like giving birth to the offspring directly. Ovoviviparous means it can lay eggs and also it can give birth to the young ones directly. So that's those are the terms you all have to remember. So oviparous is laying the eggs, and viviparous is laying like giving the birth to the child directly, and ovoviviparous means it can do both the things. And yeah, that's it about this. And double circulation is starting from amphibians, and it is completed in the aves and mammals. So using that, you can the characters common to reptiles, aves, and mammals. See here, keratinized structure and internal fertilization, and also parthenogenesis. Can we do parthenogenesis in reptiles, mammals, and stuff? Parthenogenesis. No, right? Parthenogenesis is not always possible. And marine life, we can't say marine life for all the things, because not all the animals are living in the marine and humans and stuff. Like not all the animals can't live, no. So we can't take that answer. So the answer is A and B. Okay, here is a question of fungi. So we can touch all the parts. So statement correct regarding mitosis and meiosis occurring in the life cycle of the fungi and plants. So let's go for the fungi chart. Hmm. Fungi, where do we have fungi? Fungi, see here. We have the chart of fungi now. So, yeah, see here. Here, aquatic organisms can be seen in phylum Chytridiomycota, and also in Ascomycota we have aquatic organisms. But in the other two we have terrestrial. Really sorry. Okay, so after that, we have the second character, nutrition. So nutrition, most of the Chytridiomycota are decomposers or parasites, and then mostly the zygomites are saprophytes, and some are parasites. And the Ascomycota, we have parasites, symbiotic, and decomposers. Basidiomycota, we have major decomposers, and some are symbionts. Okay, remember these terms, some, not all. Here they are mostly. And cellular organization, most of the Chytridiomycota are multicellular and they are cenocytic. Cenocytic means they do not have septa. And mycelium is cenocytic, but they become aseptate when in the reproductive cells. The zygomycota can become septa, can have the septa only in reproductive cells. And Ascomycota unicellular or filamentous or multicellular. And Basidiomycota is having what? Septa and are dikaryotic. Okay, dikaryotic mycelium is present in Basidiomycota. And cell wall, everyone is having chitin. And asexual reproduction, Chytridiomycota is having flagellated zoospores. And zygomycota is having sporangiospores. Okay, they are produced endogenously. Sporangiospores are produced endogenously. And in Ascomycota, we have the conidia. Here, all these are produced exogenously. And then there is no anything mentioned for Basidiomycota because it doesn't have. And in sexual reproduction, again, Chytridiomycota is producing by zoospores. And in Zygomycota, they have a structure called zygosporangium. There are like 7 features of zygosporangium. You have to refer those things. And in Ascomycota, we have asci. Asci is producing ascospores, and ascospores like eight ascospores would be produced for each asci. And then we have Basidiomycota, which is the fruiting body. This is for the basidiocarps are produced by Basidiomycota. So this is the sexual reproduction thing. So you have to remember these things separately as sexual and asexual. So yeah, meiosis takes place in zygosporangium of the mucus. Is meiosis taking place in the mucus? Yeah, right. Meiosis can take place. Meiosis takes place during germination of megaspores of Selaginella. That is also correct. Mitosis taking place during formation of male gametes in flowering plants. Is it mitosis which is taking place? Sorry. Second statement is wrong. Meiosis never. It should be what? When producing megaspores, it's not the meiosis, it's the mitosis. Okay. Mitosis takes place during the formation of gametes. Yeah, during the gamete form. We can be correct because the spore production is having what? Spore production is always uh, done by mitosis or meiosis. Spore production of spores, mitosis or meiosis. Tell that. Production of spores. It's meiosis, right? Very good. It's meiosis. Production of gametes are mitosis. So meiosis takes place during germination of megaspores of seed plants. Okay, not the germination. It should be, yeah, that's wrong. Not the germination. It should be the production of megaspores. Mitosis takes place during formation of male gametes. Yeah, that's correct. Mitosis takes place during the formation of endosperm of the flowering plant. That is also correct. Meiosis doesn't take place in the ovule of the cycles. That's wrong. Meiosis takes place. So the answer is what? A, C, D. And now, so we are almost done with, like we touched each and every part, and I think there are 95 questions in this tute. Okay, for the unit 3, you have 95 questions. So is it clear for you all up to this? Like, was it like a revision for you? Like, even though the time is not enough to do the revision like completely, because we did the revision session earlier also, like revising the whole syllabus using the tutes and stuff. So she's just done to go through the questions and all, no, just to see all the things to remember, revise all these stuffs mainly. So is it clear for you all? You all have any suggestions? Clear, no? If you all have any doubts or if you all want to like, yeah, Ethiopians can do internal fertilization. Clear, no? So what you're supposed to do now is you all can do the whole tute. Try to do the whole tute, like unit 1, 2, 3. Try to do all the questions today itself, and I'll upload the scheme tomorrow night. And then for unit 4, we'll have another day to do the unit 4. and then do the biocheck tools. I'll upload the biocheck tools once again in the group. So do that also. So we'll give the scheme for biocheck today. So y'all can correct the biocheck tools with that. And yeah, you all can try the essay questions as well. So let's wind up the session then. If you all have any doubts, drop me a message. And is there anything to ask? Clear. Okay. No doubts, no? Barakallah. Okay. So let's wind up the session then. Assalamu alaikum wa rahmatullahi wa barakatuhu and a very good morning. Thank you for joining.